So good morning, everyone. Can you hear me well? Yes? Yes, yes, great. No, thanks, Sebastian, for the kind introduction. And thanks for, yeah, it's a pleasure for me to be here. It's my first Sentronic user meeting. I hope not the last one. And yeah, so I'm here today to give an overview of what I call it here, agile PAT concept to showcase drug product uniformity. So good. So basically, I will first give a short introduction about PAT at Roche Synthetic Molecules Technical Development. I will go for the proposal, basically, that I want to show here. It's to eliminate thief sampling. I will explain in a minute what is this about. I will show one real example of one of our portfolio products, and also I'd like to have a link to continuous manufacturing. Finally, closing with some remarks. So what do we do at Roche Basel about PAT, technical development, synthetic molecules? Basically, we are a team of 7, 8 colleagues, PAT experts, more or less, let's say, and we have, I would say, 5 focus areas. We focused on continuous manufacturing, both drug product and drug substance. We have drug substance applications of PAT, drug product, as well as particle design, which is the link between drug substance and drug product. So what do we do? Basically, in our team, we first always give support to troubleshooting during process development. So we give support to our colleagues from formulation or for drug substance synthesis. We start with risk assessment of the PAT procedures. Whenever we see a case where we can implement the PAT method, we start with a risk assessment. We have PAT feasibility methods, then when successfully, we go to development and hopefully to validation of those PAT procedures. But we also try to find and evaluate new PAT technologies or new innovative solutions. And in my case today, I will focus a bit on that, which I believe it's a bit innovative, at least from our side it was. And this is about a proposal that we had end of 2022, beginning of 2023. And there was a question in our team that we wanted to support more batch drug product development. And the way we saw that they were doing for many, many years, and not only in our company, many companies, blend uniformity was always using this final blend, thief sampling, HPLC. We see that there is a lot of improvement there, and we saw, okay, with PAT, we can support that. So our proposal was a bit, at the beginning, challenging for management, I would say. It was a new change. We wanted to implement for all suitable synthetic molecule products a PAT-based approach to replace PU thief sampling and to show batch uniformity during the whole process design until PPQ. Focusing here only in development, let's say, because we do not target that for commercial, but at least we need to show that until PPQ, process validation. So the expected outcome of this proposal was, of course, for us was to eliminate the complexity of the thief sampling, which is a manual sampling method. We wanted to improve as well the operator safety. Basically, we don't need to open any container. We wanted to lower the risk at the PPQ. What we have seen in the past in some of our products, we were about to fail blend uniformity during PPQ. And the main reason was because of the thief samplings, which is very bad, right? Also, we wanted to increase the process knowledge. Typically, by BU thief sampling, we get a final point, meaning we have information of the final blend, and by using PAT, we can generate a lot of more data and valuable data. And also we wanted to improve robustness, meaning that the more data we can generate during development, the more we can make sure that our processes are robust. Just a quick comparison between thief sampling and PAT. There is these columns here. I'm not going to that detail. I would like to look at the figure on the right-hand side. As you can see with the figure, you can already see the benefit. On the first image, we have an operator handling a lancet. Doing the thief sampling, you can imagine the time they spend, the difficulty, the complexity. It's also something which is dependent on the hands of the operator. Not every operator has the same agility to take the samples. Instead, if we go for this picture, nice picture here where we have this central pad BU attached to the blender, we can basically rely on an instrument taking the data for us. So not only the safety, but also the quality can improve or improved basically by using PAT, and also the reliability on the PPQ and the cost. You can imagine we don't measure any sample any longer. The spectrometer does this for us, and we can have it almost real time, if not real time. We don't have to wait to the lab for a couple of weeks. Good. So the objective and the scope of this proposal, it was a bit beyond blend uniformity itself. So our objective was to replace the BU-thief sampling during development until PPQ is mentioned, and using NIR for blending, we proposed to monitor all blending steps, and not only that, because we provide BU NIR data, of course, but we wanted as well to complement this data together with tablet analysis. In this case, CU analysis using PAT, either NIR or Raman, any of these technologies to complement this PU plus CU data to ensure that the batch at the end is uniform, which is our goal. And of course, we minimize the analytical burden, as mentioned before, for both PU and for both CU. So I've been working on blend uniformity, not as many of you, not so long, a couple of years, I'd say, 9 years now. And I always had the challenge from my colleagues from formulation that, well, it's good that you can monitor blend, right? You can do a data analysis. If you don't have any quantitative method, you could at least do PCA, for instance. You can show the scores trajectory with the time. It's nice, dynamics, but at the end, my colleagues told me always, I like it, but I want a number. I want to have something similar to HPLC. I can't go ahead with a score number. So I said, yes, for that we need a quantitative method. But of course, during development, we have a very limited amount of drug substance. And before I met my friend that I will show in the next slide, it was almost impossible because you could have calibration blends of 1 kilogram, the smaller blenders that we had in the past in my previous company. So it was not easy, not to say impossible, to generate so much material during development. So in 2020, I made my friend Centro Blender, which is shown here. You know it. And for me, that was a great discovery. I wasn't aware about this instrument. Basically, with this Centro Blender, we changed the way we're doing our methods during development at Roche, synthetic molecules. Basically, the idea is that we prepare very small blends, 30 to 50 gram blends. We prepare 7 calibration blends, and you can imagine with this amount of calibration blends, taking an example, 20% drug load, we need around 70 grams of drug substance, which is something, if I compare to what I was asking 5-7 years ago, I need 2 kilograms of drug substance, and told me, you're crazy, no, forget about that. So now with, let's say 100 grams, we can go for this approach, and basically not only we blend the calibration, prepare the calibration blends, we make sure with the central part that at the end the calibration blends are uniform, so we can use them to prepare a PLS model, for instance. So once we have this, we can easily go to a larger scale. We have a PLS model, we can go to larger scale, we monitor a pilot scale, a technical scale-up, anything, and we can then predict the amount of API in the final blend, which is something that let's say 2 or 3 years ago, we could only give scores. Now we can give a value, which is what they want. Not only that, we can use these small calibration blends. We can compress them. Let's say we compress them in style 1. We take 30, 40, 50 tablets, and we measure them using NIR or Raman, so we can develop easily as well content uniformity methods. I didn't mention before, but we are not using any HPLC here. We rely on the amount of API that we have on every single calibration blend, so I know exactly the composition of each calibration blend. And of course, we develop PAT methods for content uniformity, and then when it goes to a large batch, we can do stratified sampling, and we can, let's say, in our case, we target to measure more or less 120 tablets per batch. Stratified sampling, we provide CU data. So now we we can generate this data, but we wanted as well to give acceptance criteria for our colleagues. How can we make sure that the batch is uniform? Basically, we followed a simple approach. For blend uniformity, all predictions, at least for the last rotation, 15 rotations, should be between this threshold, 90, 110. It's up to us to define this one. But RSD, which is typically something that they use in HPLC, we target the same. It should be below 5% at least. And for content uniformity, basically, we use acceptance value, which is a standard approach for content uniformity. So I'm talking about the final blend. That's right, but this is a typical example. We have 2, 3, 4 blending steps plus the compression. And then basically what we do right now is we provide data, qualitative data analysis on the first blending steps. We can show a score trajectory also for us to make sure that there is no issue on uniformity. But still, for the final blend, we give a value on final API content, relative standard deviation, as well as the tablet content uniformity analysis. So this changed a lot, the way our colleagues from formulation see PAT now. And yeah, this is something that I wanted to share as well. This is our central blender here. We used at the beginning some bottles that we had, yeah, we had some colleagues with nice ideas, and we wanted to mimic as much as possible the way we mix our products, our formulations in large scale. So we came up with this idea about these 3D printed containers. Basically, they try to mimic similar geometry as a container, as a blender. We have 2 different sizes, 200 milliliter, 500 milliliter. So we can have a calibration blend between 30 and 150 grams. As I said before, we want to go to the lower end, 30, 50 grams, but sometimes we have formulations where we have 3 dosage strength. They are dose proportionals, and if we want to make sure we can use the same calibration blend to tablet 3 dosage strength, we need a bit more of API, but still it's quite low. And yeah, I have here a small video. Yesterday you had a chance to see it in real life, but I thought when I saw, okay, it's pretty cool that we can put it here. If it works. Yeah, it works. Yeah, this is an example with our 3D printed container. Yeah, and basically now I will show now an example of a real formulation from our side, but now we are using it more and more. So a real example here, we have formulation. Now it's late phase development. It's a 20% drug load, and we have 4 blending steps. Between the second and the third, we have Rayleigh compaction. And we have 3 dosage strength. This is what I mentioned before. For that, we had to prepare calibration blends around 100 grams. So this was the pilot molecule for this replace 6 sampling proposal. We did 7 calibration blends, as mentioned, 140 grams of drug substance. With that, we could have 1 BU model for the 3 dosage strength plus 3 PAT models for content uniformity. We did that. I mentioned before we have this molecule. This process includes Rayleigh compaction step in the middle. For the calibration blends, we basically load all excipients and API, and we used this Rayleigh compaction strategy or thing that they have in the style one, basically to mimic a bit this physical variability that we can include with the Rayleigh compaction, and we used weight as a reference. So this is an example. Of course, we wanted to make sure that all calibration blends are homogeneous. So we followed this cost trajectory first, and then we can use the last spectra to build a PLS model. So yeah, PLS model, everyone knows it here. But most important is that we started this in beginning of 2023 for this project. Since then, we have manufactured 27 batches, 12 screening DOE, 7 clinical resupply, plus 8 primary stability campaign batches. So for all those batches, we haven't done any BU-FIB sampling. We've used only PAT. And just as an example, this is the primary stability campaign, which was a bit challenging, not from PAT point of view, but from management. They said, are you sure you want to use only PAT? And we say, yeah, now or never, right? So we did that, and as you can see here, this is an example. This is the final blend of the 8 primary stability batches. I did a zoom in the last 15 spectra. As you can see, all the predictions are well within the 90 and 110%, and we provided as well content uniformity data. I'm showing here comparison between BU, NIR, and CU. NIR, only the first batch what was a bit different about BU and CU. However, the results that we got, basically all batches were within the criteria that I shared before with you. So we could basically use only PAT data. And actually we are using this data for the dossier, for the filing of this project, no HPLC anymore. So we had very good results with that. And that's why when you show something which works well, other colleagues, other products, they want now to use this approach, and we are trying to do this more and more. That was nice, and it's nice, to be honest, but then I'm working as well on the continuous manufacturing line from our side, and I got a question myself. Okay, looks great for batch, but what about Conti? Is there a way we can use that to support continuous manufacturing, meaning we are using small amount of material? And just before going that way, let me introduce you a bit the mini-batch continuous direct compression we have at Roche. It's now under qualification. Basically, we have 4 losing weight feeders when we can feed in parallel the API and the excipients. This material goes into a so-called mini-batch blender. It's a plowshare mixer, and we target to mix around 1 kilogram of product. We call it mini-batch. It can be called as well mini-blend. Maybe you know it by that name. Once we blend this material, we have a weight verification. In this case, we make sure that all the material that was blended, was fed and blended into the blender, leaves the blender, meaning that there's no material left in the blender. We have this weight verification. And then we have the opportunity to have a sampling pour, or we can use PAT using stratified measurements while the mini batch is going through the pipe. And then we have a material diversion point here. We divert mini batches. They go to the tablet press when blend assay, blend uniformity meets our criteria. When they don't, then they can be discharged to waste. Then they go to the tablet press, where we have a continuous tablet manufacturing using the basic functionality plus PAT in the feed frame. And then we have transfer to coating. So here we have few advantages from what we see, a benefit of this mini batch continuous direct compression. Not going into that details now. Just to show you how does this look like in reality. So this is more or less the mini batch genesis. We start feeding. We target about 2 minutes of feeding. Then we bring the material. It goes to the blender. We blend. Target about 2 minutes. We have a high shear mixer there. After 2 minutes, we empty the mini batch. And at this point, we have the diversion point. When the mini batch has good blend assay, good blend uniformity, it goes to the tablet press. When it doesn't meet the criteria, it goes to waste and quarantine. So then it goes to the press when successfully, and we start the tablet compression. But thing is that we are sequentially producing mini batches. At the moment, the first mini batch leaves this line here, it goes to the tablet press. We start the feeding of the second mini batch. Sequentially, we're producing every 6 minutes, more or less, 1 mini batch that goes into the tablet press. And at the end, they meet all together in the tablet press. And I put this difference here because at the end, we know that there is some degree of mixing between mini batches. Good thing here is that we make sure that all mini batches have the desired quality before they go to the tablet press. Good. So this is an overview of PAT on our mini batch direct compression line. We have, I mentioned before, the option to measure BU after blending. BU at the point of compression, you see here in this picture, our feed frame in our tablet press, in our line, and also, of course, we have the option to measure at-line measurements with NIR or Raman transmission. So I focus here on the NIR feed frame measurements. I know there are many experts on NIR feed frame measurements here, and yeah, let's see how it goes now. So basically, we want to take those measurements to support resident stem distribution for every formulation in this line, and to support us into defining the frequency of sampling of tablets. We want to evaluate as well. We can use it to evaluate the potential segregation we might have for a given formulation. It's a direct compression, so this might be something we want to have a look at. We want to evaluate as well if we can see differences between mini batches. And of course, this is the last point before compression, so we want to make sure that at the point of compression we have the right distribution of the API. So how do we use, or how we are using it right now with what we have at Roche? So we have starting using the same, and this is an example, and I can give the name because it's a naproxen, it's a model compound, and we use this formulation for dust measurements in our line, so I can freely talk about that. We basically prepare 7 calibration blends again, naproxen 25%. This case we did BU-thief sampling, let's say, like this. Yeah, we took those 50 grams and we took 10 samples, and then we measured those samples with the Sentronic FO. But then one could ask, and don't look at me, Steve, I think, of course, the challenge, how do we build reliable PAT models in the fit frame with outline samples? And yeah, this is what we got at that time, and we wanted to have a model ready for the dust measurements. So what we did, we have this very nice old-fashioned device here. This is a rotating disc here where we fill the entire sample that we took, all those 10 samples that we take here on the right-hand side. For each small calibration blend, we took them, we place one by one into this rotating disc, and you can see the speed now. I hope. Actually, it's moving. If you can see that, it's very slow, right? But this is what we got at the time, and with this little amount of material, basically it's naproxen. You know, naproxen has a very nice NIR fingerprint, and we could basically have a good calibration model, of course, of a static or outline measurements like this. And then what we did is to use this model to predict a real measurement on the fit frame of our continuous manufacturing line for the dust measurements. And yeah, to be honest, this is what we got. This was about 45 minutes of measurements in the fit frame. I said 25% of naproxen. Here we are around 5.3 as an average. So when I saw that with this little amount of material and effort that we had at that time, I was surprised, really positively surprised that we were able to measure so good in the fit frame. I know there are better options to calibrate models for the fit frame, but at least this is what we got at that time with this little amount of material. And yeah, this is basically my second last slide that I wanted to share with you just now with some remarks here. I keep again the picture of the central blender. This changed the way we are doing drug product development and PAT at small molecules at Roche. To be honest, together with the Sentronic BU, we can now basically say that we are replacing thief sampling at all. We are targeting not using it anymore. Even for PPQ, for the process validation, we target to go only with PAT data. And yeah, these benefits of the agile PAT methodology, we call it agile, just to give it more fancy, right? It's not only helping the drug product development, but can also be useful to find new ways to develop PAT models for our continuous line. Yep. And of course, this is not only me working on that. We have so many people. I just place here some names. Some of them you know. Dominic Kumli, I guess you know him well. We're working together on that. And yeah, if you have any questions, yeah, I will be kind to answer them. Thank you very much.
Not sure. It seems to be working. I'm trying to not move around too much. Thank you very much, Manel, for this nice starting presentation. There is time for 2 or 3 questions. Are there any? Katrine? Yes.
Thank you for the presentation, Manel. Are you using any of your methods in commercial production, in GMP, or is it only for developing your products?
We use only for development until PPQ. We do not target that for commercial. Of course, we have, at PPQ level, we have, let's say, a reliable method we need to prove. And those could be used, for instance, when we want to transfer from one site to another to make sure that blend uniformity is given as well.
So can you share the reason why you have not entered your methods into commercial production?
Well, basically for our formulations, typically we do not have blend uniformity as a release or IPC method for our commercial products. That's the main reason. If we had that, of course, we should go with more a validation approach, and then we would use that anyway.
So you don't think there would be a benefit even though it's not a release method?
You know, we started discussion, exactly discussion, when we started proposing that, and they said, if we are not doing today anything about blend uniformity, even for HPLC, we don't do that, and why should we invest more on that? That was the main answer. I said, for me, yeah, it's less work, let's say, because it would be nice to really bring it to the commercial landscape, but yeah.
Thank you.
No, welcome.
Thanks, Katrine. Steve, sampling or sample size, sample volume, or something else?
Wait for it.
Oh.
Yeah, one thing, that the dust method where you were cycling the powder around on the disc,
yeah.
Did you have to optimize the speed of rotation?
I wish I could, but it was given. So it's a very old device that we have, and we can't really regulate the speed. This was actually what you saw here was the maximum speed we could achieve. So
Really?
You can imagine. That's why I was saying, yeah, I expect, yeah, a lot of questions on that, but yeah.
Interesting. Thank you.
By the way, this Centro dust, when Uwe has shown the Centro Mag this morning, we had an accessory which was a Centro turntable, which was simply a dish rotator. It was pretty much the same back in 2004, 2005. Any other questions? Thanks again, Manel.
Thank you very much.
Great talk, great starter.